We're about to start our class.
The next time.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. I'm pleased to chair this session. This happens to be the last session of scientific session of this International 15th International Symposium on Chemistry, ISNPC. So there are 5 presentations in this session, each of 20 minutes. And if you all are interested to be on time and finish your talks in about 18 to 20 to 80 minutes, and need 2 or 3 minutes, last 2 or 3 minutes for the Q&A. If you forget, I'll be here, inshallah. I will remind you 2 minutes before time. So the first speaker for today is Dr. Hassan bin Asif. He has recently he's a recent graduate of ICCBs Institute of Chemistry here at ICCBs, and currently he is an assistant professor at Auditorium Medical College, Isra University, Karachi. His research interests include biochemistry, molecular biology, bioinformatics, and artificial intelligence, and he has several awards and honors to his credit, including Best Oral Presentation Award at Islamabad, and has awarded several publications as well. So I welcome Dr. Hassan bin Asif for his talk on prevalence, dissemination, and antibiotic resistance associated with soil, environment, and terrible.
Assalamu alaikum, all of you. First of all, I would like to thank the organizing committee for inviting me and giving me an opportunity to present my work at this international conference. Second, I'm thankful to the chair of the session who introduces me. So, the talk title is Prevalence, Dissemination, Antibiotic Resistance Associated with Soil, Environmental, and Oxide. And this is, of course, a little slice of the big pizza from my PhD work, which I am going to represent here. First of all, before moving towards the work, we need to assess, or we need to know what is anthropoxide and why we choose anthropoxide as our microorganism for study. So as you can see, anthropoxide is basically a two-sided coin, one having a villain characteristic and the other side of the coin is having a heroic characteristic. So the normal or the introductory part is they are gram-positive bacteria, but they are no more regarded as grass, generally regarded as safe. They are catalase and oxidase negative. They belong to lactic acid bacteria family, and they are the indicator of veterinary and human resistance surveillance system. Also, they are the indicator of hygiene standards for fresh and bathing waters worldwide, and bioindicators for metal pollution like AS, Hg, and copper. On the other hand, they are also the nosocomial pathogens. They are the third most commonly isolated bacteria from nosocomial settings. They cause meningitis, bacteremia, UTIs, and endocarditis. The virulent factors they have, or the mobile genetic elements they possess, they have aggregation substance which help in adherence. They have the capability to form biofilms. They have gelatinase, hemolysis, hydrolase activities as well. They have toxin and biogenic amine productions on their credit as well. And they have plasmids, transposomes, and insertion sequences which make them very much hotspots for acquiring and disseminating antibiotic resistance and virulence traits to other genuses as well. On the other side of the coin is they also serve as probiotic by their anthocyanins or their polypeptide proteins in several veterinary applications like piglets, chicken pets, in several food systems like dairy. We can isolate them from dairy products, meat, fish, fermented vegetables. They have several medical applications including GIV disease, skin infections, and anti-MRSA, anti-VRE, anti-inflammatory. This circle in the middle shows the antibiotic resistance genes they share among different bacteria, like for example Staphylococci, Listeria, Streptococci, and Lactobacilli. They give and take different type of antibiotic resistance and virulence genes, not only among the same genus but also to other genuses as well. So this is why we choose Enterococci as our study microorganism. Now, the rationale for choosing soil for the isolation of these bacteria is soil is basically a hub of contamination, micro pollution from every area of the society, from animals and birds' manure to sewage spills, which is common in our society. Human activities are also the contributors for polluting the soil. Hospital discharge is of course one of the major pillars in this area. Industrial waste, and all these sum up to the soil contamination, which concluded in human health hazards. That's why we choose soil as one of the unnoticed sample site for our study, because this has not been under study previously. So now coming to the sampling site, we, as you know, that Karachi is a metropolitan city and the local government has divided the Karachi into 18 towns. So we planned our study in such a way that we sampled the soil samples from every town of the city. All the 18 towns were sampled equally for soil samples, and we present Karachi as a town model system for this study. Coming to materials and methods, soil sample collection done. Then we isolated the enterococcus by selective enrichment of enterococci in different concerned media with 6.5% NaCl and 45 degrees Celsius, because this is the selection criteria. Then selective and differential streaking on diaspirin and SBA plates to pick a single colony and purify the strain. After that, we did antibiogram analysis and virulence factor enzyme assay analysis by different broad dilution and agar-based methods. After that, we identified the related genes which are related to antibiotic resistance and virulence in enterococci by multiplex PCR strategy. And after identifying the isolates which are multi-virulent and multi-antibiotic resistant, we compared them by web PCR for their intra and inter-species variation and connectivity. So the results, as you can see, we have found a huge number of enterococci, different species of enterococci from soil. We have total 372 isolates from 500 samples. We have collected 500 samples from throughout the Karachi, and out of those 500 samples, we have 372 total isolates having the proportion, major proportion contained by Leafaceum followed by Leaficaris. This shows the phenotypic isolation of bacteria, and this shows the multiplex PCR for the identification using 16S rDNA gene for genus specification and superoxide dismutase gene for specific species identification. This diagram shows the antibiogram analysis. As you can see, we have done the antibiogram or antibiotic resistance analysis by agar dilution method, by diffusion method, and by broth microdilution method. We check the MICs by broth microdilution method as well. And as you can see, we have planned our work in such a way that we included major antibiotics of all the classes, from protein synthesis inhibitors, from cell wall synthesis inhibitors, and from DNA replication inhibitors, and included them in our study, those antibiotics which are commonly used to treat human infection as well as animal infection in our society. So we have found a great proportion of resistance, majorly from meropenem, the imipenem class of family, then lincosamide and high-level aminoglycosides and high-level glycopeptide resistance is also shown. As you can see from this graph, this shows MIC of soil isolate as compared to the clinical isolate. This is a reference clinical isolate we use in this study. And as you can see that the soil isolate has a much higher resistance to the aminoglycoside we checked as compared to the soil clinical isolate. After that, we have performed multiplex PCR for the identification of a specific antibiotic resistance genes for those isolates which are positive phenotypically for those antibiotics, including aminoglycosides, erythromycin, the macrolide family, PBB5 for ampicillin, which is beta-lactam, and transposon 916, which is helpful in transmitting or exchange of antibiotic resistance characteristics from one organism to another. Tetracycline resistance and ciprofloxacin resistance genes were also checked by multiplex PCR strategy. After antibiotic resistance profiling, we checked our isolates for phenotypic virulence determinants, including hemolysis assay, lipase activity, biofilm production, gelatinase assay, and urease assay. As you can see, these pictures shows the phenotypic characteristic of the selected anthropoxide we have taken into our study. After getting the phenotype positive, we ran multiplex PCR for identifying the basic genes which are involved in different virulence determinants. So these are the biofilm associated genes. ACM stands for adhesion to collagen. Efaceium carries more antibiotic resistance as compared to E. faecalis. E. faecalis carries more virulent determinants. But selected virulence characters can also be found in efaceium as well. So these are biofilm associated genes, adhesion to collagen, enterococcus surface protein, and adhesion to surface tissues by efaceium and E. faecalis. These are the gelatinase operon genes, the serine protease, GAL-E, FSR, A, B, C. So we have concluded the whole operon in those which are positive for gelatinase activity. After that, this is the cytolysine genes, Cy-A and Cy-L, which help in the this is the promoter region and it helps in the cytolysine activity. Anthrocin genes, anthrocin A, B, P, AF1097 was also evaluated. This is anthrolysine A gene, which is associated to cytolysine characteristic. Lipase related genes in efaceium and E. faecalis. And this is the hospital associated marker IS16. It is to highlight here that we have found more than 50% are isolates carrying these IS16 insertion sequence 16 marker, which is an indication that they came from or their origin is from hospital discharge. So this is one of the eye-opening results of this study, and these are catalase. These are for catalase G. After multiplex PCR of antibiotic resistance and multi-virulent enterococci, we have selected some of the major multi-virulent, multi-antibiotic resistant enterococci and check the intra and interspecies variation by box PCR. Box is basically a repetitive motif. First of all, identified in the Streptococcus pneumoniae, but it is commonly found in many other bacterial species and genuses as well. So we exploit this technology to make an intra and interspecies variation to find out the biogeographical evaluation or evolution of our microorganism. We have collected the microorganisms from all over the city, so we want to know their biogeographical evolution. So as you can see, the box PCR analysis after dendrogram construction showed 4 different plates or 4 different groups and different isolates obtaining from different areas of the Karachi city, including the ATCC strains and some clinical isolates as well as a representative in this study. They all are sorted out into 4 different groups, and each group shows that these isolates have their origin from a single point. So by this we can conclude that the isolates we have from all over the Karachi, first of all, they link to different biographical locations, and they, as they carry I16 gene, so it is more possible or probable that probability is more that they came from hospital discharge as well. Conclusions and recommendations, as I have shown enterococci, this is a well-established nosocomial pathogen. It is associated with various diseases and it is virtually resistant to all clinically useful antibiotics we use in our study. Presence of antibiotic resistant enterococci in soil opens a new route for the exchange and dissemination of resistant genes, not only among the microbes but also to the food chain. So it is recommended that a regular monitoring and surveillance must be taken to avoid these pathogens enter into the food chain of humans. It is also recommended that hospital waste must also be disposed of very carefully, as we have seen much of the isolates carrying hospital associated markers, the nosocomial lineage in our environmental enterococci. These are our little contributions from this work. The work has been published in Microbial Pathogenesis and also as a book chapter in Intact Microorganisms book. These are some selected references, and the financial acknowledgement. This project was financially supported by HEC to my supervisor, Dr. Professor Saeed Awad Ali. And thanks to all my lab fellows. These are my lab fellows, including my supervisor. And me and one of my friend who started under the mentorship of our supervisor this project. And so this is his intellect, his guidance, and his brainchild project, which supported me and which makes me enabled that I am in front of you here today. Thank you.
Thank you, Dr. Sham, for a very nice presentation. Nice work. We have time for one.
Very nice. Do you find policy
Not yet, but this is need future plans, because doing a global surveillance is like a consortium work, we have just touched, like I have just touched the soil area. There are other environmental aspects as well, which are being under observation in our laboratory. So after getting in touch with all the environmental aspects, including like soil, air, water, sewage, animals, and birds. So by gathering the data from all over Karachi, then we can jump to the global surveillance.
This is my Our next speaker is Dr. Yasuddin. Welcome him for the next talk. He is Associate Professor at the Government Environment Center for Research, ICBS. And his research interests include computational biology and bioinformatics. And he's been working in this area since 2012. And he's the author of and co-author of about more than 60 publications. Main focus is computational biology and bioinformatics, in which he analyzes big data for solving complex biological problems. Particular focus is resistant pathogenic bacterial genomes and corresponding translated gene product. He has many awards and honors. He is very particularly the Young Motivational Scientist Award from National Academy of Science, Young Scientist, Pakistan. So I welcome Dr. Yasuddin.
First of all, I would like to thank this conference organizers as well as the chair for introducing me the chance to reflect my work, which I have been doing for the last couple of years. So, should I stay here? So, in order to start my work, I would like to start my presentation first slide is saying about why this antimicrobial resistance is global concern. You see, like this is a major problem these days. Antimicrobial resistance definitely kills, and it challenges care and control of infectious diseases, and it greatly increases the care associated cost, and it threatens a return to the pre-antibiotic era, where actually antibiotics were not working, and it jeopardized the healthcare gains for individual and society, which we have gained over a couple of centuries, and topmost is the by 2050 it is predicted that by Wellcome Trust, like resistant infections will be the #1 cause of death globally, with an estimated 10 million deaths per year. So we have to think about it right now. Some notorious examples of antimicrobial resistant bacteria are, for example, tuberculosis, which has about 440,000 new multi-resistant TB cases annually being reported. And about XDR TB cases are reported in 64 countries so far. It's a big threat to the health concerns. Similarly, malaria is getting resistant to ongoing use of monotherapies, and HIV is also giving some resistance. And notorious famous example is nicotine resistant Staphylococcus, which is nosocomial and against which many drugs for like antibiotics are failed, and so on. So the question comes in how bacteria get resistant actually. So there are multiple mechanisms. One is it may stop the antibiotic from reaching its site of action, and or it modify or bypass the target. Our work is focused on in this second mode actually, how bacteria get resistant. So in this case, most of the time bacteria camouflage the target. How it basically changes in the composition of the structure of the DNA, which leads to some mutations that may lead to new genetic products, which comes as a complicated gene products. And then the antibiotics for which actually it was made for, they were not able to actually bind with those proteins, new proteins, and then it gets failed. And one more thing which regarding with this topic is reprogramming targets. Sometimes they produce different variant of a structure of a protein. For example, vancomycin resistant bacteria is a famous example where actually it codes for new kinds of different cell walls, which is actually different than the sensitive bacteria. And the antibiotic is not actually made for this kind of new cell wall, so that it failed to interact. And sometimes bacteria actually express new alternative proteins. And those proteins are basically giving shield to the bacteria for the applied antibiotics. And sometimes bacteria gets horizontal gene transfer from the environment and then get new mutations and new proteins comes in. And in this way, they get new kind of set of protein and metabolic pathways through which bacteria can get resistance. So our work is focused on, as I said earlier, this is bioinformatics and data intensive research. So it starts with the DNA and DNA codes the information to RNA, and then RNA passes to the protein. So in between these phases, there are lots of data coming up, which needs to be analyzed by for every possible organism. So as everybody know about that, the discovery of new drug is not an easy task for early research of drug discovery, and it is expensive and laborious as well. And on top of that, the time required to bring a drug into open market is quite long, which is about 14 years. And alternatively, the identification of unique drug targets is another approach through which you can actually tackle the bacterial resistance and bring new drugs as well. So we are focused on metabolic pathways that are associated with drug resistance and those proteins which are unique to resistant bacteria only. And we try to identify novel potential drug targets among those new set of variable proteins. So everybody knows about that, like the genomic sequences actually revolutionized the biology. Biology is now considering as a more like a biological information science. And it has, every other organism is now being sequenced, and every half an hour, if you go to the LTBI database, you will find new pathogenic bacteria and other organism data coming up and updated. So this availability of complete genome sequence has led to radical changes in the life sciences, and it is a well-known fact that as many as about 30 to 40% of the bacterial or other organism genome is still being characterized as uncharacterized, for which you don't know the function yet. These are largely ignored genomic information for which nobody knows what it does. This is where actually we can find some new clues about the new metabolic pathways and new proteins through which you can bring new drug targets. So this figure is even higher for archaeal and eukaryotic genomes and relatively larger genome of bacteria. And those remaining genes for which there is no function assigned, they are categorized or labeled as in genome sequence data as conserved hypothetical genes or non-characterized or unknown or orphan genes. There are those keywords which you can find easily in any complete genome sequence of organisms. Moving on further, like if you know about those genes for which you don't know the function, but you can predict the function by using the data. So one of the characteristics of the gene network is basically the proteins which are involved as like the most connected proteins. They are considered as a drug target or hub proteins. So through that, if you know the cluster of genes with known function in which hypothetical proteins are being characterized, that means it is basically functioning the same way as the cluster is. So network characteristics can also be used for the protein interaction analysis, so that you can bring new PPI inhibitors. So how it workflows actually, so you start with the complete genomic sequence of every possible organism which you want to study, and then perform these steps in cycle and repetitively, and then bring up some new and prioritized shortlisted drug targets through which you can use, you can actually perform some structure-based studies as well, and then go for molecular docking and simulations to bring new drugs. But in order to find the function, you need to perform this whole pathways analysis, evolutionary research, for example, the phylogeny analysis, and sequence analysis, and PPI data analysis. This all steps actually go into a kind of work frame. So through that, I will now share some examples which we have done so far in our lab, and a few examples are shared here. So one of the topics I covered is the Pseudomonas Aeruginosa, which is very, very notorious for having resistance to almost every possible antibiotic available against it. And we perform whole proteomic analysis. By doing so, actually we started with the proteomic data of every possible strain of the Pseudomonas Aeruginosa. It is 68 at the time when we studied. and in which one of the PA01 is the most the resistant form of Pseudomonas Aeruginosa. And through that, we actually covered all proteins into and clustered them into a group that is called cluster of proteins, and then find out like which proteins are basically essential and and playing roles into the regulatory pathway of the unique metabolic pathways of only the bacteria, not the host. So this kind of steps have been performed, and then later on the gene network analysis by using protein-based network analysis was done, and we applied, as I said earlier, the subtractive genomics analysis on the core proteome of Pseudomonas Aeruginosa and how proteins were identified, and proposed them as novel drug target. They were 8 in number, and if I give you an example of what kind of a number of data we were, tackle in this work is basically there were total number of strains at 68, and about total number of protein sequences were there that much. And you see again, this 30% of these complete proteome sites were actually unexplored previously. So this kind of tagging and this kind of labeling of such data is good to further future drug discovery downstream analysis. One more example from our work is basically the subtyping of hepatitis C virus by using bioinformatics and mass data. And in this work, actually, so hepatitis C virus is famous for having so many subclasses, subvariants, because of the mutation rate is so high in that. So in order to, actually bring a drug combination for the therapy of hepatitis C virus, you need to know the type of the hepatitis C virus. So in order to, actually find hepatitis C virus is very, very challenging, because every time you find new variant coming up in hepatitis C virus, because it's RNA virus, very quick mutations. So one of the ways you can find through that, like you can completely find every possible subtype complete proteomic data, and then globally align them, and then find the consensus sequence, and then find the occurrence of most occurring amino acid sequence into that, and then bring up a global consensus sequence data, and then you can clip them by using proteolytic kind of enzymes, and then get the fragments of the stretches of the peptide sequence globally consensus actually, and then theoretically calculate the mass values of it, which is possible because the amino acid mass spectrum libraries are very, very commonly available. And then you can find the occurrence of it through this work has been done through 2 algorithms. One is the flu align, through which we align every possible sequence of the type of the hepatitis C virus, and flu jest, which actually gives and establishes the uniqueness through a comparison with the masses of all proteolytic peptides across all protein and strain of the virus. So that if the uniquely present peptide stretch will give you the idea that particular strain is basically the label due to that presence of these kind of stretches in there. And in order to further validate it, you can perform mass spectrometry. And if you find the theoretical calculated value is found in the spectrum as well, it correlates with the data, and then you can confirm the type of the virus and stuff. So this kind of work actually have been done in our lab also. And moving on further, I would like to share a recent work which we have conducted with a database development of unique metabolic pathways of infectious pathogens. You see, not everyone is basically expert in bioinformatics and genomics data, so they may like to work on that. And the problem is that there are lots of databases available, but there is a lacking of the unique metabolic pathway data. There is no kind of such database before. So because we have been doing such work already for many pathogens, so why not we can curate it even to kind of single database so that it can be available for everyone. So this is the motivation of this work. So this database is about to be published very soon, and this is the first page of this website would be very soon. It will be uploaded and available for everybody. And what it does actually, it is expected to expedite the process of discovery with drug target because it contains only the database. It is a database for only unique metabolic pathways, not anything else. So anyone who actually doing some kind of drug discovery research, they may go and find organism-specific unique metabolic pathway data through which you can find the new drug target, and in that you can perform some kind of new drug discovery study in that. So what are the objectives of this kind of database? Basically to identify the unique metabolic pathways of all existing pathogens. This is the goal actually. So far, we have got about 5,000 pathogenic data available in here, but we are still moving on further. And we also want strains to be included in that. So, you can imagine how comprehensive this database will be. And to identify new and unique therapeutic targets and to automate the process of drug discovery. And through that, we perform some kind of coding, which actually being developed here, and it can automate the process of identification of targets. And same here. So what is the benefit the scientific community working in the areas of drug discovery sciences, particularly they can get greatly benefited by this database by directly targeting the unique proteins, not only the all, and new drug targets can be identified also. Companies like pharmaceutical and biotechnology, they can easily use this database for their projects that might be strain specific or species specific. And portability of this database will be made available by downloading facility. Everybody, because it is curated one time, and then you actually download it in one day and then use it for further analysis. And future direction, we would like to integrate with different biological databases such as NCBI and Unitort, so that you can directly get this sequence curated also. And that database will be linked with the programs such as BLAST algorithm, and we can perform on the fly these comparison and social media accountability. So this is again the first page of this database, and this is how you actually drop down menu and they can get index organical specific pathway information, and then you can further go down and you get the protein information as well. This is how it looks like. It's going on further, something back. So with that, I would like to summarize my presentation today. The computational biology methods help to bring new knowledge. Yes, there are lots of data available which must be ignored. You may need to find some new information out of this data. That is the computational biology part being done. And it is helping to bring due attention to the unexplored or uncharacterized genomic data. This is the major advantage of major goal of this study. And it helps in proposing the possible drug targets against the infectious pathogens, particularly the resistance one. So, हम लोग का जो major काम है, we already always work on mostly resistant pathogens. But there is always challenges. Every now and then we are getting new data, and new data is being updated very rapidly. So, how do we use it in a quickly manner and then find the new information quickly? That is a big challenge. And sometimes the methods are not as appropriate. We need to define and design our own pipelines. That is a challenging environment. And sometimes some molecular biology and the biological method, the biological phenomena are still not clear for some diseases. And also that's the science. We still need to be explored. And with that, I would like to acknowledge all of my lab members who work with me together. I work in Palmani Center for Research, and I acknowledge their excellent facilities providing us for our labs, and some of the funding agencies which are funding our work. With that, thank you so much.
Thank you, Dr. Vyas, for your excellent work. The floor is open for questions. A couple of questions.
Like you said, that you're exploring the unique metabolic pathways, right? So how you are going to determine that it is specific for this certain strain of bacteria? Or
that's the data which we have available for every possible strain. And if we talk the terminology unique in this database sense, is basically unique to the pathogen only, not with the host. It's not common with the host. The first thing we need to find is the uniqueness towards the pathogenic strain, and then we can compare that every strain. For example, one of the work we have done with Staphylococcus aureus, with every possible strain, we found only very like one or two metabolic pathways we perform specific only to certain kind of drug resistant pathogenic strain. So the data is available. For example, if you know about the CAT database, for example, this is randomly scattered there. So what you need to do is just scatter the data, and then using the coding, you can calculate, you can find the linkages with them. And if you simply plot a heat map, you can easily find like one side you have every strain, another side another strains, and then you can find like which one is common and which is unique. So this is how we do it.
this uniqueness is referred as the uniqueness between host and bacteria, right?
Yes.
So in that case, how much really will be that it is unique from the normal flora as well?
Yes, that's a very
Because
good
that is
question.
one
I
of the
agree.
very major concern
Yeah.
when you're treating the animals.
Most of the time we get the reviewer question, the same question which you have asked actually. So there is another database is available for the microbial flora, which is normally found in the humans, gut microbial flora. So if you actually you can use that database also to compare your found unique metabolic pathway. So if they are common in there, you can easily exclude it. And there are some unique metabolic pathways which are not common in microbial gut flora. So this is you can do it through database available for the microbial flora specifically. Yes, we do that.
My question is that you found unique metabolic pathways, and you also found some kind of attractive targets. Have you worked further based on the metabolic pathway,
Yes.
target some specific protein or enzymes?
I agree. Very good question again. You see, if you talk about database, so we only show you the cataloged, unique metabolic pathways and specific proteins. But those proteins is still taking role in the unique metabolic pathway of the bacteria, but may still be common with the host. So, which is actually the next stage to find those proteins which are actually found only specifically to the bacteria, pathogenic bacteria. So I showed one example of Pseudomonas Aeruginosa, for example, and we have published it so far, like the metabolic pathways, 10 metabolic pathways, and the 8 prioritized drug targets. So this is I would say, limit of my work actually. This is too much data to be handled, But this is open for those people who are actually working in structural bioinformatics. So those 8 prioritized drug targets, out of these thousands of proteins, they are prioritized as a drug target. So that means the shortlisting is being done, and people who are actually really want to perform drug discovery based study, their work is basically the long journey is basically now shortened, and these 8 prioritized drug targets are open for the structural bioinformatics to be applied these structural based methods. So we did not do that. We limit up to there, and we move to another pathogen. And in this process, we got so much of data to be curated, which we curated through database of unique metabolic pathways.
So now that we will continue this discussion with the break. So we are moving on to the next video. Thank you very much for a very interesting talk, and there is a gift for you. Speaker is online from Indonesia. Dr. Alfreda Novi, this county, Associate Professor at the Chemistry Department, Faculty of Science and Technology, University Erlangen campus in Indonesia. She is an advisory board of Indonesia Chemical Society since 2016. She is author and co-author of a number of publications in the field of organic chemistry with H-index 11 and I-10 index of 15. Her publications have been published in many peer-reviewed journals, and she has research interests in natural product chemistry with organic synthesis. So I welcome Dr. Alfreda Novi on the talk some non-polar compounds from Session-2 aqueum step 1. Dr. Alfreda.
Okay. Do you see my PPT, my presentation? Hello?
Yes, we can see.
Okay. Do you see the PPT is moving the second?
Dr. Finda, your screen,
Not
your
yet.
slides are shared.
Not yet.
Kindly start the presentation.
Wait a moment.
Slide soon.
Describe your slide.
Okay. Okay, good morning. First of all, I would like to thank the committee for allowing me to present our study in the seminar. This time, I will present my studies about some compounds from 2 species of
Dr.
CCK.
Alfinda, we cannot hear you. Please go ahead.
You don't hear? You don't hear my voice? Hello?
Dr.
You don't
Alfinda, can you
my-
hear me?
Yes, I can hear, but you don't
We
hear my
cannot hear
voice?
your voice.
Oh.
Pakai ini mungkin Bu, pakai kuping sudah ya.
Right. Now you can hear. Hello? Hello?
Yes, Dr.
You don't
Alfinda, we
hear-
can hear you. Please
Oh,
go
okay.
ahead.
Okay, thank you. Okay, good morning. First of all, I would like to thank you to the committee for allowing me to present our study in the seminar. This time, in this occasion, I will present our study about some compounds from 2 species of Syzygium. This is our university at Indonesia. Okay, Syzygium is one genus, belongs to Myrtaceae family. Many studies have been carried on the species of this genus, such as Syzygium aromaticum, Syzygium comini, Syzygium quinens, Syzygium jambus, Syzygium malescens, and Syzygium samarangiens. From some publication, we summarize that the most reported secondary metabolites from this genus are terpenoid, steroid, chalcone, flavonoid, lignan, tannin, phenylpropanoid, and hormone. And bioactivities reported from this genus are anti-diabetic.
Excuse me, ma'am, we cannot see your slides.
Oh,
Kindly share again.
no? Do you see?
Yes, now it's clear.
You can see the moving in the next slide or no?
Here is slide number 3.
Okay.
Yes, move. Thank you.
Okay, I continue. And bioactivities reported from this genus are anti-diabetic, anti-fungi, anti-inflammatory, antibacterial, antioxidant, cytotoxicity, anti-HIV, anti-diarrheal, anti-atherosclerotic, and antivirus. Syzygium aqueum is a plant native of Indonesia and Malaysia. This species we know well through the edible fruits. This one, which is sweet and a little sour. The second species that I will present is Syzygium polycephalum. For the last species, our study is still ongoing, so only a little information that will give in this presentation. The previous researcher who studied the species have been reported the isolation of tannin and flavonoid compounds from the leaves of the species. And some of these flavonoids have already reported that anti-diabetic activity and cytotoxicity on cancer cell line. The aqueous and methanol extract of the fruits were also showed cytotoxicity on some cancer cell lines. Any researchers had never reported the phytochemical compounds about the stem bark of this plant. So we work on this part of this plant. The procedure of isolation is presented in this slide. The sample is a stem bark that we ground to form a powder and then macerated with methanol at room temperature. And the methanol extract obtained was evaporated and then partitioned with N-hexane and ethyl acetate. So we had 3 fractions: nonpolar, semipolar, and polar fraction. After checking with the LC to see the profile of the compounds in its extract or in its fraction, we decided which fraction will be separated. The separation was carried out using chromatography, such as vacuum liquid chromatography or VLC, and column chromatography, gravity column chromatography. Several separation is needed to arrive in the pure compounds. From the LC chromatogram, we are interested in separating the N-hexane fraction. In the first separation using VLC, we obtain 19 fractions. Not all of the 19 fractions produce directly pure compounds, and we choose interesting fraction to separate more until the pure compounds got. You can see the scheme in the slide. Compound 1 is the simple phenolic compounds, namely 2,4-tertiary phenol. Compound 2, 3, 4, and 5 are steroid compound, which are butyrospermol palmitate, butyrospermol, cytosterol, and beta-cytosterol. And compounds 6, 7, 8, and 9 are triterpenoid compounds, which are oleanolic acid, alphytolic acid, a derivative of oleanolic acid, and macellinic acid. Ethyl acetate extract is also potential fraction to be separated, even though the profile is less rich than N-hexane fraction. In the first separation using VLC, 10 fractions were obtained, which only 3 of them give the pure compounds, which compound 10, which is bentonelic acid, compound 11 is another derivative of oleanolic acid, and compound 12 is arginolic acid. Furthermore, these 12 isolated compounds were tested for anti-cancer activity using MTT and XCT assay. The MTT assay was carried out at Gadjah Mada University, Indonesia, while the XCT assay was carried out at University of Technology, Malaysia. Not all compounds undergo the same assay because it depends on the availability of the compound sample and also the availability of the laboratory who do the test. The overall test results are shown in this table. The most important results shown by the beta-cytosterol with IC50 3.43 microgram per milliliter. The interesting result also shown by alphytolic acid and arsenic acid. Although the IC50 value, these compounds were still above the IC50 value of doxorubicin as a positive control, it can be said that these compounds have a potency as anti-cancer agents. Due to the good IC50 value, an in silico experiment was carried out to see the potential of these compounds as anti-cancer. In this case, as inhibitor of human estrogenic 17B hydroxysteroid dehydrogenase type 1, or its SD17B1. This enzyme provides biosynthesis regulation of active estrogen and stimulating the development of breast cancer through the cell proliferation. Molecular docking was initiated with determining the superposition inhibitor its SD70B1 complex conformation. For this experiment, inhibitor or ligand native of this enzyme was testosterone. So, in order to have a similar structure to the ligand native, the compounds that will be analyzed in silico are beta-sitosterol and oleanolic acid. This slide shows the molecular docking result of the candidate, which is beta-sitosterol and oleanolic acid, and its SD70B1. This compound shows closer on the pocket area of the enzyme active site. The correlation also shown through the grid score or binding free energy, with the energy trend of C1 enzyme is stronger than C2 enzyme, and C2 enzyme is stronger than ligand native enzyme. This shows that C1 and C2, which is beta-sitosterol and oleanolic acid, are promising as an inhibitor of the SD17B1 enzyme. Key residues that have stronger free energy in its complex, you can see in this slide. For a ligand enzyme, there are 5 residues. For C1 enzyme, there are 6 residues. And for C2 enzyme, there are 4 residues. Key residues play the interaction with inhibitors on the SD17B1 packet enzyme. Leucine 149 and proline 187 were highlighted in the inhibition mechanism of ID70B1 enzyme. From previous research, we know that leucine 149 residue plays a role in stereodiscrimination and modulates GST level in breast tissue, while proline 187 is a hydrophobic and aromatic residue that contributes to the main thermodynamic force for binding mode. Hydrogen bonding analysis is the main interaction between ligand and protein. The hydrogen bonding number parameter shows the number of hydrogen bond interaction between ligand and protein. The result of the analysis showed that there are more hydrogen bonding in C2 protein compared to ligand native protein and C1 protein. Furthermore, experiments were carried out to determine the hydrogen bond occupation. This experiment showed that C2 protein complex has two strong its hydrogen bonding categories to the presence of the hydrogen and carboxyl group, which playing an important role in ligand protein interaction at the atomic level. We continue to other plant, other species, namely Syzygium polycephalum. Several phytochemical studies have been conducted on this plant. And the ethanol extract of leaves showed strong antioxidant activity. This is 2-saturated fatty acid, some terpenoids, and steroid were isolated. The whole fruits were reported to contain anthocyanin with a strong antioxidant activity. From the bark and stem bark, and also the cortex, it was reported the presence of gallic acid, ellagic acid derivative, and flavonol glycoside. The stem bark and bark showed strong antioxidant activity, while the hardwood were the potential as antifungal. Due to the limited report regarding to the compounds contained in each part of this plant, the study was performed to explore this plant, including the seeds, fruits without seeds, and leaf. The isolation procedure was the same as that used in the previous species. In this work, we separated from the seeds. For this experiment, we separate the seeds from the whole fruits. And from the seeds, we isolated two phenolic compound, which is gallic acid and methyl gallate. From the fruits, we isolated trimethyl citrate and dimethyl citrate. And from leaves, we isolated a derivative of ellagic acid. Antioxidant activity was performed using ABTS method. In this result, we see that only the gallic acid showed the good potential antioxidant more than Trolox as a contrapositive. Whereas all extract obtained from maceration and partition, also tested by ABTS method, and only ethyl acetate extract of seed showed weak antioxidant activity, whereas other extract did not show antioxidant activity. For conclusion, 12 secondary metabolite compounds were isolated from stem bark of Syzygium aqueum. These known compounds have not been reported yet from any parts of Syzygium aqueum, although there is not a new compound. Some isolated compounds have a high toxicity to cancer cell line. For example, beta-sitosterol for HeLa cell line, alpha-tolic acid for P47D cell line, and arginolic acid for HeLa cell line. However, all compounds had lower activity than doxorubicin, which was used as a positive control in this assay. All calculation in silico approach suggests that candidate inhibitors, which is beta-sitosterol and oleanolic acid, have a worthy potential to inhibit the HSD17B1 enzyme. 5 compounds from the fruits, seeds, and leaves of Syzygium polycephalum have been identified as dimethyl citrate, trimethyl citrate, methyl gallate, gallic acid, and a derivative of ellagic acid. The antioxidant test using the IBTS radical showed that gallic acid had better antioxidant activity than control Trolox. I'm very sorry that not being able to attend in this seminar in Karachi, Pakistan because of the pandemic. This is not over yet. Hopefully, we from Indonesia can attend to upcoming seminar, maybe next week, we hope. And I would like to thank you for all students in our laboratory and also the member of our research group, which provided satisfactory research and also have created a pleasant working atmosphere. And thank you very much for your kind attention.
Very nice work and nice work. The house is open for questions. Time for questions.
I just want to ask which program, docking program used for this study. Docking program.
I'm very sorry, I cannot hear well your voice.
I just want to know which docking program you used. Can you please tell me the name?
I have to see because I have a student who. Yes, I will inform after I have a student who is mastering to do this part of our research. So I can give you the information.
No problem. You don't need to look at it. You know,
Okay.
I just want to.
I'm very sorry.
Thank you.
Any questions? If there are no questions, let's end of the agenda. Can you hear me, Dr. Ganda?
Yes, I can hear, but not very clear.
Oh, thank you very much. I want to thank you for
Okay.
your participation.
You're welcome.
In person in Karachi in the next conference.
Okay, thank you very much.
We move on to our next speaker, Dr. Muhammad Imran Malik, Associate Professor here at the HJ Research Institute of Chemistry, ICCBS. His research interests include polymers, synthesis, characterization, and applications. He has a large number of awards and honors to his credit, so many publications, and with very high impact factor exceeding 200 with an index of 16. And he has also written a book, three book chapters, along with a book titled Advanced Separation Techniques for Polymers. So over to you, Dr. Imran.
So, as you can see, today I'm going to talk about molecular imprinting based technology for basically purification of natural compounds from the plant extracts. This work we have started just recently in our lab. And the reason is interesting, I want to share. Actually, as the Honorable Chair already mentioned, I belong to this institute. And now it has diversified a lot, but when I joined here, it was only organic. And now, and basically focusing on natural products. So when I joined here, I am polymer chemist, more focused on sensitive factors of polymer and their applications. And when I meet my colleagues, we talk about everything but not science. That is something I was missing, that I want to have something our lecture to colleagues so that we can talk science. So at that time I started thinking about how I can talk science with my colleagues. From that point on, with these thoughts in my mind, I see this view a lot in the laboratory. So this is a metal column, its height is almost like this. So basically, my colleagues and the students are looking for novel natural compounds and other natural compounds with a potent medicinal activity, and they want to purify that. From the plant extracts, of course, and of course, plants a big source of bioactive compounds. So this procedure, it takes around maybe, if you know this extract and you want to separate some specific organic natural compound from that, this may take from weeks to months. So at that point, there are lots of other things, lots of solvent consumptions, activity as well, because this works only on the crystal polarity. At that point, I thought, can I facilitate this step somehow including my science in this? So for that, I knew at that point molecular imprinting is something very special. So I started thinking about it in back in 2013. I started thinking about it, how I can contribute in natural product. I started thinking about it, and then this is basically a molecular imprinted polymers. That is, different square nature, like when it is an antibiotic interaction or very specific interaction. So we would like to prepare artificial receptors for a specific compound. See, this is how it works. It's the template is the target molecule, and these are the functional monomers. These functional monomers interact with the template, and they become pre-polymerization complex. And then once they make this complex, we add some cross-linker, we have multiple double bonds, and then polymerize. And it makes a solid matrix, matrix around this template. And then we move the template, and then that solid matrix we have the cavities that have very specific size and specific function group at that particular position. So once we expose this material to this compound, it traps this compound potentially compared to two things. One is this interaction is generally non-covalent, can be covalent also. There are some reports, but it should not be weak enough so that the polymerization is separate from that. And it should not be strong enough so that when we want to move it, it will become difficult. So in principle, we always prepare non-infected polymers, also non-infected polymers exactly same, just to complete this template. So that all will have all the functions in there, but randomly separated. So of course, this does not have any competition with the color development for the new compound, for now. So we need a template, we need a compound. But of course, if you want to enrich some compound from the all that this matters. You want to enrich it, you want to verify. So for this, we take monomerization as a water compound here. This is naturally plants and many. So we use these different monomers, these different functional monomers here for that, and this the composition is the class linker, and then we check the absorption capacity of using these different monomers. And at the same time, we make NIPs and we check its absorption capacity. And then the most important thing is the entry factor, which is the ratio of MIP and NIP absorption capacity. So see for these two ones, we found so this thing about this monomers taking the highest absorption. Then next time we mix these things and make a combination of that and this added well behind it product here. So this basically has a composition of 1,1,5, and this gives the integrity factor of 4.4. So this is how we try that to mark and move forward. So here I want to have attention of my colleagues who are working on the computational chemistry. You see, we use the monomer combination as I am supporting the down here, and you can imagine student must have done maybe 20 or 30. And if you can guide us where your ex-situ computation chemistry, which monomer or combination of monomer is most suitable for this particular type. That will reduce our workload required very much, and this will be a very contribution. So next thing, once we make it, we want to characterize this MIP. As I said earlier, MIP and NIP are the same. All the elements of template is the next one. So we want to see it more the same, but if you want to tell there's some peculiarities of the MIP which we have explored. So here you see this is NIP, washed MIP, and unwashed MIP. Unwashed MIP means it has the template in there, and from there we remove the at first look, they look very similar, we're happy. And then, but if you go into detail, you see this peak is a bit broader. This amino functionality, this is the stretching peak of amino, and this is the basically a bending peak of this 1461.5 of the amino. So these two peaks are a bit distant. So this shows that this MIP, which is washed, it has some interaction. Amino groups of the MIP of the polymer has some interaction with the template. And then we see this is the MIP and this NIP. These are same images. If you look at from the low resolution, they look exactly same. But once you go there, at high resolution, you see their pores, which are not present. So it means it has a large surface area compared to NIP. And these results are confirmed by BAT analysis also. You see, BAT shows that the surface area of MIP is almost 6 times more than the NIP. And this is about thermal degradation profile, which tells us that actually these two polymers are same. So that's what we like to see. We like to see they are same, but at the same time they're very different. Before moving into MIP, because later I will discuss more about this much is a log and this much is a dotted plot. For that, these are the two methods. We built two methods for the concentration of RA. One is UV. So see it has a specific absorption 326 and we made an elevation curve. And as you can imagine, for complex mixture, these UVs can overlap. So we just separate it by HPLC, and then you see this is like elevation curve. So later, whenever I say this much is absorbed and this much is not, we have used these two methods for the determination. So now come to the performance of this component. We have optimized the extraction performance. So this is a different pH. We use different pH. What we need to know is this blue one is MIP and red one is NIP. What we need to note about is the difference between MIP and NIP. So you see here, small difference, which is comparatively less for the NIP. So this low pH is basically the pH which is low, which should be used for extraction. Second thing is the concentration of MIP. So concentration of the RA or analyte. So as we increase concentration, you see the dropping capacity increases. And of course, this means there are more, there are still available cavities for trapping this analyte. But when we reach around 500, after that, we don't see any improvement. So it means that all the cavities available are already trapped by the analyte. And here's the time. So you see, it needs around 60 minutes. After 60 minutes, we don't increase that. So these are the optimum extraction conditions. This is the concentration of RA and time. We did some 30 studies, and we see from this sketching plot, it shows that there are two groups of binding sites. That is what we like to see. One is low affinity binding site, and one is the high. While NIP has only one kind. Absorption kinetic profile show that it follows pseudo-secondary kinetic profile. So till here, you see things looks quite good. We have a difference between MIP and NIP, but major test would be how much our MIP is selected for the other compounds. What is the difference between these two? For that, here we use different compounds there that should be present in the extracts. So you see these different compounds that are structurally similar, and here you find a difference, but not for these others. So this is the imprinting factor of rosmarinic acid, and the other imprinting factor you see of that. And it's basically selectivity factor. This is a ratio of the analyte, the other analyte, and the RA. Then you mix this with the different concentration of the interferent, and you see that there was not a big difference in the efficiency of MIP for our MIP for the extraction of RA. Moving next, once we have till here, we have just extracted the rosmarinic acid selectively, and then next we need to dissolve it from that matrix so that we can have pure compound in hand. So we have to optimize the desorption protocol. For that, we use different solvents, and you see ethanol found to be best one with 80% recovery. We use the different amounts of the solvent, not the difference, and 10 minutes are enough for this. So these are optimized desorption. Using the optimized absorption protocol I described earlier, and optimize these organ protocols, we try to see the reusability potentials. And you see, after 5 cycles, the potential, the efficiency of the MIP is reduced by. So, and the little bit decrease in the efficiency may be due to absorption of RA, better permanent state problem, reversible state problem. So, how is the real test? Putting all this on the to the plant extract. So, this is the good plant extract. Where bad PLC, it has many different peaks. I don't know what's in the peak, how many peaks. We confirm this peak because of the retention time, and then we actually LCMS that this is a bigger part. We treated this with MIP, and you see here under same conditions, we should look at the relative intensity of the peaks, and you see this peak, R peak has reduced to large extent compared to other peaks. Similar thing we did with NIP, in which there are not specific capital, and you see there is not much difference in the relative intensity of oils. So this selectively, actually extracted RA from that transaction. Then we dissolve it from the MIP and analyze with the same method, and you see this is almost pure, slight amount of this one. Otherwise, it is almost pure. And once we treated this one, this of this one, and see the multiple peaks. So by NAP, these were not separated well. By MAP, separated well. And all this took 1 hour of absorption and 10 minutes of absorption. So this brings me to the end. So I hope I'm able to convince you that MIP technology can be used for extraction of smart compounds from organic natural compounds of medicinal important potency, and the multi-function monomer technique would be better compared to the single function monomer polymer, but it requires a small amount of template. So see, it has no competition for the novel compounds, but for the compound which is novel, these are generally accepted, and we need these compounds for some other activities. It's easy scale up method can be easy scale up. Further application of MAP, you see, MAP based degradation of dyes. What we actually do, we concentrate the dyes at one point, and then the efficiency of the degradation catalyst enhances by many folds. One of my students is working on this. Another student is working on electrode modification of MAP for using the biosensor for electrochemical electrochemical. It can also be used in a stationary phase of chromatography and SPE. These are the applications we would explore in future. As I said earlier, that I started reading about this topic in 2013 on the background, and reading and writing after in 2019, we were able to publish a review article about it in a prestigious review journal, in fact, in documents, and then this is the first publication of this series from our lab. This is my team. I appreciate all of them for being brave to accept new challenges, to starting new research domains in my lab, and of course their endurance, their hard work enabled me to be here to present the work. This particular work is done by Sonia. It was her emphasis, and she recently graduated. Ali is also working on the same project, and he will graduate hopefully for his PhD in this year. I'd like to thank Professor Shekari and Professor Shankari for providing me compounds and extracts, and Dr. Homa for helping me in interpretation of some of it. And obviously, a big thanks to all of you for being here at second last lecture of the last session of this conference. It required a real practice.
Thank you, Dr. Ram, for your nice presentation. Now to open for one question.
Yes, may I see the table of the first few slides?
Yes.
Yes. You asked to reduce your workload
Yes.
by involving computation.
Yes.
But this calculation requires quantum mechanical stuff, and quantum mechanical calculation takes a lot of time. But other thing that the kind of based on this data, only interaction which could be calculated to find out which is the best partner for your medical. But you didn't mention the medium, I mean solvent, gas phase, or in which medium you are carrying out
Yes,
this.
sir. There must be a solvent involved.
Which solvent? Because the solvent.
There are many solvents. That is what we want you to decide, to tell us either methanol, organic solvent, acetone, or acetone.
Polar to non-polar.
Whatever. Our template should be soluble in this, and our polymer should be insoluble in this. So this is what is question for you, please.
Basically, you are asking a lot. It is not only the solvent, different composition also.
Composition, yes, of course, the glossary.
Thank you, Dr. Imran, for a very nice presentation. We can continue our discussion in brief as well. And post the conference is ending, so we have plenty of time for that. Thank you very much. And there's a good for you. Presenter is Dr. Antibiogram, Associate Professor at ISNPC-15, and his research interests include the theoretical and computational studies, in particular application of advanced molecular dynamics simulation, including quantum mechanical effects, to complex chemical, biochemical, and biological macromolecules for the investigation of their structural and dynamical properties. He has many awards and honors to his credit, published around 50 articles in the field of theoretical and computational chemistry in the Journal of International Review. So I welcome Dr. Day.
Thank you. Thank you, Chair, for inviting me. This is the title of my talk, which is establishing rationale of simulation. This study is based on the selection of a compound from natural source and analysis of the emission potential against tumor pathology. This is the compound, which is a session-2 natural compound isolated from a plant, Polygonum barbatum. And the compound was characterized as phenol acid of the 7 of the 1,2-methyl propylene and compound plate. Using different spectroscopy technique, the compound was also established to possess good anti-proliferative and anti-metastatic properties. And this work was published by my colleague Dr. Ayaz in 2017. After that, we discussed to move further to work on the target-based studies for the evaluation and potential of this compound against gelatinase, which is involved in metastasis. This slide could give a kind of insight into the role of MMP2 and quinine, which is called gelatinase in metastatic process, how metastasis work. What happens when the create a signal enters the cell, a complex chain of events within the cytoplasm is occurs. These events kind of lead into the cell nucleus, where the transcription of gene regulating cell cycle rotation and cell growth are stimulated. One of the proteins produced during this cell activation process is MMP or matrix metalloproteinase. When a tumor cell kind of separates from the normal cell, they secretes MMP or matrix metalloproteinase, which degrades the extracellular matrix, which is a collagen. And after this, the tumor cell migrate to the blood and blood or lymph vessels. This is all the metastatic process. What is MMPs? They are generally excited by fibroblasts, neutrophil, macrophages, and tumor cells. They degrade all kinds of extracellular matrix proteins. What the extracellular matrix? It's a kind of three-dimensional network of extracellular macromolecules. ECM or extracellular matrix, it forms an essential support structure for cells. It controls communication between cells. MMPs, they are also involved in the cleavage of cell surface receptor and chemokine inactivation. The activity of MMPs is precisely regulated by endoproteinases and PIMPs, tissue inhibitor of metalloproteinase. They play a crucial role in controlling the local activity of MMPs. Moreover, they are also known as matricin. Essentially, they are calcium dependent, zinc containing antioxidants. They belong to a large family of proteins known as matricin superfamily. They have a common structure and consists of propeptide domain, catalytic domain, and hemopexin-like C-terminal domain. And this terminal domain is linked to a catalytic domain by a peptide domain. As I said, it's zinc-containing antipeptidases. That's why it has an integral zinc ion into the active site of these enzymes. And they comprise of various types, but our focus is gelatinases, and it consists of MMP2 and MMP9. It's also called gelatinase A and B respectively. At the early stage of immunogenic evolution stage, MMP2 overexpression occurs, whereas MMP2 is involved in the advanced stage of cancer. They are also associated with neurodegenerative disorder. Both enzymes collectively serve as engineering factor and promote tumor externalization with the resistance of vascular growth factor, which results in metastasis. They contain three fibronectin type 2 redundant domains that contain bindings of collagen and gelatin. These are the binders of the MMPs. Basically, these binders coordinate to the zinc ion, replacing the water molecule in the active site of the protein. So there are different ones like hydroxamine, hydroxyurea, and hydroxyurea. So based on these, there are a number of drug molecule or drug-like molecule which binds or coordinate to the zinc ion of the protein of the gelatinases. But what happens? All these drugs, they have some undesirable side effects, which are represented by most common unavoidable adverse events, such as musculoskeletal syndrome. And that's why none of these inhibitors have successfully entered market. Although they have undergone various preclinical and clinical stages. For example, this is Bioinformatics. This is the drug which is the first synthetic MMP inhibitor to enter clinical stages to treat cancer, but at stage 3 they were taken out from the market. So this will be attributed to the lack of selectivity towards the specific MMP subtypes that might be responsible for this failure. Therefore, it is a challenging task to achieve this selective inhibition since most MMP isoforms have similar structure and function due to the similar active site, zinc-containing active site. So this is a zinc-containing active site, catalytic site. You see, they both are similar. The active site is similar. It contains 3 histidine residues and one water molecule, and zinc binder replaces the water molecule to form a protein complex or adder. So to overcome this challenge, there is another side which is called S1 prime binding pocket, and for between these two enzymes, between MMP2 and MMP9, they have different locations. So we call that the synthetic compound. It has zinc binding motif, just like in this case hydroxamine, but the isolated compound, which was reported by my colleague, it does not have zinc binding motif. That's why it is not going to bind with the zinc ion. So based on this criteria, we kind of selected this compound for our study, and our objective to dock this compound to other side because it does not have a zinc binding motif. That's why it's going to bind. It was like hypothesized to bind with the other side, that's on prime side. So to solve our problem, we adopted this techniques and dissociation. Is a computational tool to observe structure and based on different mechanisms. There's a lot of technical stuff. I'm not going to detail, but the application is so vast. So this is the kind of overview of the method we apply, which is called molecular modeling and dissolution protocol, which involves the first step, the selection of the crystal structure and the preparation of protein ligand complex using molecular docking. But the most kind of difficult task to kind of parameterize or to obtain metal ligand information. For this purpose, we kind of implemented quantum mechanical calculation at the density function level of theory. After that, The whole is the complex was already neutralized, and the typical MD submission protocol involves minimization, hitting, and integration in production MD. So what happens when we simulate a molecular system, complex molecular system? We obtain trajectories or the huge data, and we analyze this trajectories or the data. We apply different analysis tool, but I'm going to kind of present only the characteristics data here. So these are the kind of plots and the theoretical data we obtained so far. The first trivial analysis or the preliminary analysis is to kind of get an indication how they can behave, how they can perturb the structure of the protein after binding. Based on RMS analysis of the backbone protein, both backbone patterns of the protein, you can see in both cases after the binding of the ligand to the protein, the RMSD got kind of got contrasting, because in this case the RMSD got increased and in this case RMSD got decreased. So it has a clear indication that ligand is going to kind of perturb the structure of the protein. When a ligand binds to a protein, the question arises of the stability of the complex. But as for the stability as well as the deviation of the structure, in both cases we can see that as the simulation approaches, what happens? A single stable conformation is obtained. This color code indicates the RMSD or the type of the conformation or single type of conformation obtained during the simulation. RMSF analysis. It is also called temperature factor or B factor, which can also be obtained from the experiment and from the simulation or from the theoretical calculation can also obtain from this. These are the residual fluctuation of the protein of the MMP2 and MP9, and you can see higher fluctuation in the region between 100 and 112 and as a prime site after the inhibitor binding. In this case, what's happening? S1 and active site region remain unperturbed after the inhibitor binding. But there are some residues between 80 and 90, which will be kind of called as structurally important region, but they are not a part of S1 and active site region. This is a kind of very typical plot and what happened after the inhibitor binding. What's happening here? That this is the collective motion of the atoms of the residue of correlation of the collective motion of the atoms and residue. And what's happening here? After the inhibitor binding, the conformation space. This is the kind of inhibitor free protein. This is a complex. In this case, also inhibitor free and complex. The conformation space of both enzymes got completely changed. These were the typical analysis, but based on those analysis, we could not figure out the kind of complete information how ligand is behaving after binding to the protein. So what we did, we performed PCA. PCA tells about the essential dynamics which is responsible for the protein function or protein inhibition after the ligand binding. So here, these are the kind of, this is called SPE plot, and based on the plot, we calculate traces of covariance matrix. And you can see after the inhibitor binding, the value got increased, and the total summation of eigenvalues exhibited the motility of the system. Then the system is quite flexible, which indicated the RMSD data we calculated earlier. In this case, what's happening? This value, there is a kind of little difference between these two values. However, the trend of the SPE plot is quite similar. Based on this data, we again calculated the RMS analysis, because in the previous RMS we were not able to identify the functionally important region of the protein after the ligand binding. And based on this RMSD, based on this data obtained from the PCA-based RMSD, we can see there is a fluctuation or there is a kind of flexibility of the protein after the inhibitor binding, which could not be possible to achieve in the typical RMSF when we perform for the MMP9. This is a very important tool to find out the conformation responsible to extract out the important data for the inhibitor binding. This is called free energy landscape plot. Based on the plot, we attempted to extract different conformation of the complex, and based on this, we were able to find a stable and transient state of the kind of complex, and we can see in case of inhibitor free, it was like 3 conformation. Afterwards, it got reduced. In case of MB9, it got increased in 3. And moreover, we were able to kind of find out the important residue which could be involved in the hydrogen bonding with the inhibitor. In this case, we found 2 residues, this one 83 and 84, which were involved with the hydrogen bonding with the inhibitor. In this case, there were no hydrogen bonding with the inhibitor. So these are the hydrogen bonding data. These are the 3 residues, and based on the percentage of hydrogen bonding occupancy, these 3 hydrogen bonding were dominant to make the complex stable. Whereas in case of MMP9, only 2 residues were identified to stabilize the complex via hydrogen bonding. So these are the structural information or dynamical. Afterwards, at the last, we perform free energy simulation, and based on the for the computation of binding free energy with respect to S1 prime side, because we docked our complex, we docked our inhibitor to the S1 prime side, and this is the kind of reverse method which is called thermodynamic integration method, which is more difficult to perform compared to other simple binding energy calculation methods. So these are the calculations obtained from free energy calculation, which is like a binding free energy minus 5.23 and 5.94. And then we discussed our work with my colleague Dr. Ayaz, and he performed some sort of gene expression studies. He showed that some sort of down regulation of this protein by the inhibitor, the sesquiterpene. So based on kind of our study, there were some sort of outcome, and we evaluated the role of this non-competitive inhibitor sesquiterpene as potent anti-metastatic agent by targeting these gelatinase expression by utilizing simulation. The subsequent MD-30 analysis tools and simulation gave insight to the inhibitory profile of the inhibitor, suggesting a possible mechanistic view. Computation results significantly complemented the experimental gene expression studies of both enzymes. Thank you so much. These are the acknowledgements. I would like to thanks to all these personalities, especially Dr. Balchowdhury providing high performance computing facility, Dr. Ayaz and research group to performing this gene expression study. My research group, especially with Sharif Jamal, my PhD student, this was her ample work. Thank you so much.
Presentation, and we now open for a couple of questions.
You have performed a number of in silico analysis, taking into consideration the inhibitor and the gelatinase. So, but I just found that at the end you perform only expression analysis to back up your so do you plan some further experiments to back up your data?
Session plan, we are going to kind of continue this work because establishing theoretical rationales takes a lot of time. Now we are planning to move further with kind of some sort of de novo analysis to modify this structure, which could be more potent than this one. Still, there is a kind of gap. We could not figure out the selectivity because the both are very similar, and the problem is still selectivity, as I mentioned in my slide. The problem, that's why all the drugs, they have been failed just because of similar similar identical subtypes, gene binder. So that's why S1 prime pocket or the binding site kind of could give a kind of good, let's say, inhibitor or compound. But still, I would say it's not kind of fruitful because in this case, I also obtained similar kind of data or similar binding ability over
Just in that case.
The one of my question has already been answered by you, just right now. So there's another question. Why do you have focused on the hydrogen bonding?
Yeah, now we didn't focus only on the hydrogen bonding, but I told you there was a lot of other data. We focus on the hydrophobic contact as well, because in the case of MMP9, the S1 prime property, the residue were quite non-polar. That's why they were not supposed to be involved in the hydrogen bonding. So we kind of plotted contact maps other than the hydrogen bonding, but I will show you. So the hydrophobic interaction or hydrophobic contacts play comparatively more role compared to the hydrogen bonding, because hydrogen bonding could be kind of solution, could be kind of broken or something like that. But hydrophobic in the solution, because in the living system it's water, hydrophobic cannot be like broken by this polar gradient or something like this.
Thank you, Dr. Tarek. And then for from the organizers.
Okay.
Time to close this session, and thank all the speakers for the wonderful talks, both in intro and in work presentation. Before I close the session, there are a few announcements. Closing ceremony of the symposium will be held at sharp 2:30 p.m. in the Session-2 Auditorium, and all the participants are requested to kindly be there on time. And a very important feature of that closing ceremony is the poster award announcements. And for poster presenters, those who have presented their posters, they are requested to bring 3 minutes presentation with them of their posters in the university, and at the time of the closing ceremony, so you may be fortunate enough to get the prize. So in that case, you will be asked to give the presentation, 3 minutes presentation. Okay, so now we will all proceed for lunch and enjoy your lunch, and thank you so much once again.